Hello everyone. It is my great pleasure to share our research entitled, Chromosome-level genome and methylome of vine T suggest roles for tandem duplication and CHH hypomethylation in high dihydromyricetin accumulation. Vine T is a characteristic medicinal and edible plant in China. Its young leaves can accumulate more than 37.5% dihydromyricetin, or DHM, a flavonoid with diverse bioactive properties. Although previous studies have advanced our understanding of the vine T genome and structural variation in DHM biotic genes, an important question remains: why can young leaves accumulate DHM so specifically and efficiently? In particular, the coordinated roles of gene duplication and DNA methylation have not been fully understood. To address this question, we integrated genomic, transcriptomic, and epigenomic data to get biochemical validation, to systematically investigate the molecular basis of high DHM accumulation in vine T. Our study provides 4 key insights into high DHM accumulation in young vine T leaves. First, we generated a high-quality chromosome-level genome. The genome is about 620 megabases, with 98.55% anchored to 20 pseudochromosomes and 97.4% busco completeness. Second, we profiled DNA methylation in roots, stems, and young leaves. Young leaves showed reduced CHH methylation, possibly due to lower methyltransferase expression and higher NgDML1 expression. This pattern was associated with high expression of key DHM genes. We identified tandem duplication of DHM biosynthetic genes. Overall, 96.4% of core genes and their paralogs were tandemly duplicated, increasing key gene copy numbers for DHM production. Finally, we propose a duplication-associated activation model. Tandem duplication, CHH hypomethylation, and MYB and BHLH transcription factors may work together to promote DHM accumulation in young leaves. To explain high DHM accumulation in young vine T leaves, we generated a chromosome-level genome. The 620 megabase genome, with 98.55% anchored to 20 pseudochromosomes, enabled analysis of DHM genes. Tandem duplicates were enriched in flavonoid biosynthesis. Although DHM gene families did not expand overall, 96.4% of core genes and paralogs occurred in tandem arrays. Multiple CHS and F3-5H copies formed clusters, were active in DHM biosynthesis, and were highly expressed in young leaves. In contrast, the Qing pathway gene STS showed no clear expansion. Why are these genes highly expressed in young leaves? We compared DNA methylation in roots, stems, and young leaves. Young leaves showed the lowest CHH methylation, at only 9.35%. Hypomethylated regions were mainly found in intergenic and promoter regions, and associated genes were in phenylpropanoid and flavonoid pathways. Key DHM genes, including CHS and F3-5H, showed promoter CHH hypomethylation and high expression in young leaves, whereas competing pathway genes remained more highly methylated. Promoter CHH methylation was negatively correlated with gene expression. We therefore propose a duplication-associated activation model. Tandem duplication increases gene copies, while CHH hypomethylation relieves epigenetic repression, together promoting DHM accumulation in young leaves. In summary, high DHM accumulation in young vine T leaves is driven by tandem duplication of key biosynthetic genes, CHH hypomethylation associated activation, and coordinated regulation by MYB and BHLH transcription factors. These findings provide a basis for elite germplasm selection and molecular improvement of vine T. Thank you for your attention.